Reminders on bacteriological characteristics and AST1 Professor Mokhtar Kamara. Identification of bacteria colonies by diagnostic laboratories is based on phenotypic characteristics, heavy checkmark cellular morphology, usually shown by gram, bacteria, heavy checkmark colonial morphology, heavy checkmark biochemical metabolism, heavy checkmark genome identification, molecular methods, heavy checkmark spectral profile of bacterial proteins, MALDI-TOF MS. 2. Cellular morphology, 1/2, bacterial motility types 3a. Monotrichous, mono means one single polar flagellum, e.g. Vibrio cholerae, Campylobacter spp. Polar flagella often in pairs to give a seagull appearance. B. Lophotrichous, tuft of flagella at one or both ends, e.g. Spirilla sppc. Peritrichous, flagella in periphery flagella surrounding the bacterial cell. All the members of family Enterobacteriaceae, if motile, have peritrichous flagella, e.g. Salmonella typhi, Escherichia coli, Enterobacteriaceae proteus, highly motile organism, shows swarming motility. D. Amphitrichous, single flagellum at both ends, e.g. Alcaligenes faecalis. Note, amphibians live both on land and in water. 4. Bacterial colonies of a single species when grown on specific. Morphology, heavy checkmark, characteristic size, heavy checkmark, shape, heavy checkmark, color, heavy checkmark, consistency, heavy checkmark, pigmentation. The growth rate of certain organisms are variable depending on 5. Colonial shape is determined by the edge and thickness of the colony. The edge may be smooth, entire, or irregular and serrated. If the colony is thicker in the center than the edge, it is said to be raised, or it may be relatively uniform, appearing like a disc. The texture of the colony is also important. It may vary from dry and friable, easily crumbled, to butyrous, buttery, to sticky, and the surface may be smooth, wet, dry, or granular. Some organisms produce a pigmented colony, which can aid in the identification process. For example, Pseudomonas aeruginosa, Serratia marcescens, Staphylococcus aureus. Other microorganisms produce hemolytic colonies. Group B Streptococcus, Streptococcus pneumoniae, S. aureus. 6. The size of bacterial colonies is generally uniform within a species. Streptococcus species are small, usually 1 millimeter in diameter. Enterobacteriaceae are larger and usually 2 to 3 millimeters in diameter. Bacillus species are much larger in size and usually 2 to 7 millimeters in diameter. 7. Appearance of colonies. Smooth colonies S. Smooth, smooth, shiny, and moist with 2 to 4 millimeters in diameter. Except Yersinia, smaller colonies. Rough colonies are rough, dry, irregular contours, and matte in color. Mucus colonies, M, with diameter 10 millimeters, tend to confluence, e.g. Klebsiella large. Fatty and shiny colonies, dwarf colonies, strains deficient in their metabolic chains, urinary E. coli strains, special case, proteus, agar invasion, uniform mat. 8. Smooth bacterial colonies, 9 Staphylococcus aureus colonies. Rough bacterial colonies, 10 Pseudomonas aeruginosa colonies, Bacillus colonies. Mucus bacterial colonies, 11 Klebsiella colonies. Swarming colonies, 12 swarming proteus culture in blood agar culture of proteus on nutrient agar medium. Colony pigmentation, 13 non-diffusible pigment Staphylococcus aureus versus white Staphylococcus spp. Colony pigmentation, 14 diffusible pigments Pseudomonas aeruginosa versus Pseudomonas fluorescent. Type of hemolysis, complete hemolysis versus partial hemolysis, greenish, 15. Identification of bacteria into genus and species respiratory metabolism, oxidase, catalase, protein metabolism. Presence of urease, indole production by degradation of tryptophan. Presence of enzymes, decarboxylases, deaminases, coagulase, denase, lipid metabolism, lipase, glucidic metabolism. Fermentation of sugars, glucose, lactose, mannitol, maltose, sucrose, etc. Use of citrate as unique carbon source, Simmons citrate, Christensen citrate, hydrogen sulfide production, gas production. 16. Gram-positive coccus API Galleria 17 API 32 strep API 32 staph. API Galleria, Enterobacteriaceae 18. 19. 20. Interpretive reading of the antibiogram is based on knowledge of resistance phenotypes susceptible. Manual AST manual methods 22 illustration of commonly used methods of antibiotic susceptibility testing. AST A, broth dilution test B, agar dilution test C, concentration gradient diffusion test D, disc diffusion test. 23.